Welcome to the deep dive. We're here to take those dense stacks of information you send us and, well, really dig in, turning complexity into something clear and actionable. Today, we're exploring a whole kingdom that often gets overlooked, beyond just mushrooms or that mold on your bread. We're talking about fungi. And we're going to show you just how vital, how weirdly specialized, and sometimes how dangerous they can be. We actually start with a story that honestly sounds almost like fantasy, but it underpins entire regional economies. I'm talking about this high-altitude fungus, Ophiocordyceps sinensis. You might know it as Yartsagunbu, the caterpillar fungus.
Yeah, it's probably the most valuable fungus out there. It's an entomopathogenic fungus, basically. It's evolved to hijack and kill insects.
Yeah.
And its treat value can be upwards of $40,000 for just half a kilogram. It's biological gold.
Wow. And that incredible price, it links back to traditional medicine claims, but also, remember those Chinese track athletes in the 90s?
Oh, absolutely. Their amazing performance was publicly credited, at least in part, to using this exact fungus. It's quite a story.
And the biology behind it is just as wild, isn't it?
It really is. So the cycle starts when these fungal spores infect ghost moth caterpillars, Thydorode species living in the soil. The fungus, its body made of hyphae, just grows inside the living caterpillar, eventually mummifying it. Then, come spring, this fruiting body called a stoma, it just erupts right out of the caterpillar's head, poking above the soil, ready to be picked.
That's incredible. And the economic impact, you mentioned it was huge.
It's profound. Back in 2005, I think the number was something like 8.5% of the entire GNP for the Tibetan Autonomous Region came from harvesting Yartsagunbu.
8.5% from a parasite. That's staggering.
It really is. And that value brings huge ecological risks naturally. Unsustainable harvesting is a massive problem. People are collecting it late in the season, which means they're taking the fungus before it releases its spores for the next generation.
Right. Interrupting the life cycle.
Exactly. And then you add high-altitude climate change, altering temperature and humidity.
Yeah.
It's a serious threat to this resource.
Okay, so that sets the stage. Let's get fundamental. We're talking about the kingdom Eumycota, the true fungi, eukaryotic, spore-bearing.
And critically, no chlorophyll. They don't do photosynthesis. Their whole way of life revolves around absorptive nutrition, osmotrophy.
Meaning they eat differently than we do.
Completely differently. They secrete these really powerful enzymes outside their bodies onto whatever they're eating. Could be wood, bread, or, a caterpillar. Those enzymes break down the food externally, and then the fungus just absorbs the resulting soluble nutrients. That external digestion is key.
So structure dictates function here. How they're built relates to how they eat. They come in two main forms, right? Yeasts and molds.
That's the basic split. Yeasts are your single-celled fungi. Microscopic, often roundish, and they usually reproduce by budding. On a petri dish, they form smooth colonies, kind of like bacteria, but the cells are much bigger. They're great for rapidly growing in, say, sugary liquid.
And molds are the fuzzy ones, the multicellular ones.
Precisely. Molds are made of those thread-like filaments, the hyphae, which weave together into this tangled mass you can often see, the mycelium. That's what bread mold is.
And even within those hyphae, there are different designs. Coincidic versus septate, what's that about?
It's about internal structure and flow. Coincidic hyphae, or aseptate, they lack cross walls. It's like one long, continuous tube filled with cytoplasm. Things can stream freely. Then you have septate hyphae. They do have cross walls, the septa, but these septa have little pores.
Ah, so the cytoplasm can still move between compartments, just maybe more controlled?
Exactly. Either way, though, this whole filamentous structure spreading out as thin threads creates an enormous surface area compared to the volume.
Which is perfect for that absorptive feeding. Maximizes contact with the food source.
Perfect for osmotrophy. Extremely efficient.
Okay, structure makes sense. Now, their reproduction. You mentioned a unique twist involving when the nuclei fuse.
Yes, this is really distinctively fungal. In sexual reproduction, you have plasmogamy, the fusion of the cytoplasm from two parents happening first. But the fusion of the nuclei, karyogamy, that gets delayed.
So what happens in between?
You get this fascinating phase called the dikaryotic stage, often written as N plus N. It means you have a single cell containing two separate, genetically distinct haploid nuclei, one from each parent, just coexisting and dividing together for potentially a long time.
Like roommates sharing a cellular apartment before fully merging. What's the advantage of keeping them separate like that?
It provides amazing genetic flexibility. It's a hallmark of the big groups, the Ascomycota and Basidiomycota. It lets them mix and match genes, essentially test out combinations before committing through nuclear fusion. Helps them adapt.
Very clever. And they still do asexual reproduction too, right? With spores?
Oh yeah, lots of asexual spores for just spreading quickly, things like cnidiospores, sporangiospores.
Mm-hmm.
But that eukaryotic stage is really a defining feature of their sexual cycle.
Let's dive into the diversity now. Starting with a group that's kind of unique. The chytrids. What makes them stand out?
Their mobility. Chytridiomycetes are the only major fungal group that produces a motile spore. It has a single flagellum at the back, like a little tail letting it swim. It strongly suggests the earliest fungi might have been aquatic and motile.
And unfortunately, one chytrid is infamous. The Trachochytrium dendrobatitis, B. This is the one causing amphibian declines.
Yes, B is responsible for what's considered the worst disease-induced loss of biodiversity ever recorded. It's been catastrophic for frogs and other amphibians globally. Just devastating.
How does a microscopic fungus kill a frog?
It targets their skin. Amphibians rely heavily on their skin for breathing and for regulating electrolytes, salts and water balance. Bead infects the skin cells, causes them to thicken. It's called hyperkeratinization.
So the skin stops working properly.
Essentially, yes. The thickened skin can't exchange gases efficiently, and more critically, it disrupts electrolyte transport. The frog can't maintain its internal balance, leading ultimately to cardiac arrest.
Horrifyingly effective. Okay, moving from the motile chytrids to the Zygomycetes, like bread mold, Rhizopus, they have those tough zygospores for waiting out bad conditions.
That's characteristic, yes.
Yeah.
And generally coenocytic hyphae, mostly saprophytes, breaking down dead stuff.
But Rhizopus has another trick up its sleeve, doesn't it? Something involving rice blight and bacteria.
Yes, the Rhizopus chinensis story. This is a fantastic example of, microbial teamwork or maybe conspiracy. Scientists found the toxin actually killing the rice seedlings wasn't made by the fungus.
No, then what?
It was made by a bacterium Burkholderia living inside the fungal hyphae, an endosymbiont.
So the fungus is basically acting as a Trojan horse, carrying this bacterial weapon.
Precisely. The fungus provides the delivery system. It really complicates the picture of who the pathogen is.
Wild. Okay, from that kind of complex interaction to a clearly beneficial one, the Glomeromycota. These are the mycorrhizal fungi.
Yes. Specifically, the arbuscular mycorrhizal fungi, or AM fungi. Absolutely crucial ecologically.
This is the symbiosis with plant roots, helping them get nutrients.
Exactly. It's usually a mutualism. The fungus uses special structures, aprosoria, to connect with the plant root cells. It forms these intricate structures inside the root cells like tiny branching trees or arbuscules. The fungus network acts like an extension of the plant's root system, massively increasing its ability to pull in nutrients like phosphorus from the soil.
And what does the fungus get out of it?
Sugars. Carbohydrates that the plant produces through photosynthesis. It's a fair trade, and something like 80–90% of land plants depend on these fungal partners.
Amazing. Okay, now onto the real giants, the decaryotic fungi. First up, Ascomycota, the sac fungi, named for the ascus structure.
That's right. The ascus is the little sac that holds the sexual spores, the ascodazospores. This is a huge, diverse group. Contains everything from baker's yeast, Saccharomyces cerevisiae, to valuable truffles, but also many destructive molds and plant pathogens.
Let's talk about yeast first. Saccharomyces. It's a workhorse in labs and industry.
It absolutely is. A key model organism. Reproduces asexually by budding, and there's a neat detail. Each time a bud forms, it leaves a scar on the mother cell.
A scar.
Yeah, a bud scar. And eventually, the mother cell gets covered in these scars and can't bud anymore. It has a limited reproductive lifespan.
A built-in limit. And the filamentous Ascomycetes, they have survival structures too. Sclerotia.
Yes, sclerotia. These are amazing. They're these hard, dense masses of dormant hyphae, like a fungal survival bunker. They allow the fungus to survive really harsh conditions, drought, cold, lack of nutrients. Then when conditions improve, they can spring back to life. It gives them a huge advantage over competitors. Think Aspergillus.
And survival can sometimes mean parasitism with devastating effects, like ergotism.
Ergotism, a truly grim piece of history, caused by Claviceps purpurea, an Ascomycete that infects rye grain. Back in the Middle Ages, eating bread made from infected rye led to these horrific outbreaks known as St. Anthony's fire.
What were the symptoms?
Terrible. Convulsions, gangrene in the fingers and toes due to restricted blood flow, and intense psychotic delusions, hallucinations. It turns out the fungus produces potent alkaloids related to LSD. The fire part likely came from the intense burning sensation victims felt in their limbs.
Chilling. Okay, the other major deckaryotic group, Basidiomycota, the club fungi. Mushrooms, puffballs, rusts.
Named for the basidium, the club-shaped structure where the basidiospores are produced, usually four on the outside. Ecologically, they are the masters of breaking down tough plant material like lignin and cellulose, critical decomposers.
But this group also contains some famously poisonous members. The death angel.
Amanita phalloides, yes, tragically famous. Its danger lies in its potent and specific toxins.
Specific how?
It produces several, but the main ones are phalloidin, which very quickly destroys liver cell membranes, and imanitin. Alpha-amanitin is slower, but incredibly potent. It inhibits RNA polymerase II, effectively shutting down protein production, particularly hitting cells in the GI tract and, again, the liver.
And the scary part is the delay in symptoms, right?
Exactly. You might eat it and feel fine for 6, 12, even 24 hours. By the time the severe GI symptoms start, the toxins have often caused irreversible liver and kidney damage, often fatal without a transplant.
A reminder to never eat a wild mushroom you can't positively identify.
Absolutely crucial advice.
But in sticking with pathogens, let's touch on white nose syndrome in bats. That's caused by an Ascomycete.
Yes, Pseudogymnoascus destructans. It's a fascinatingly specialized pathogen. It's psychrophilic, it loves the cold.
Cold-loving.
Yeah, its optimal growth is around 12 degrees C. It can't even grow above 20 degrees C. This makes bat hibernation caves, which are typically cold and humid, the perfect environment.
So it grows on them while they're hibernating.
Right on their exposed skin, particularly their wings and muzzle, hence the name white nose. This fungal growth irritates them, causing them to wake up from hibernation far too often.
And waking up burns precious energy reserves.
Exactly. They burn through their stored fat needed to survive the winter. They basically starve or freeze because of these repeated disturbances. It's led to absolutely massive declines in North American bat populations.
Another example of extreme fungal specialization. Okay, last group, the Microsporidia. These were confusing for a while.
Oh, incredibly confusing. For decades, they were bounced around taxonomically, often considered protists. They are obligate intracellular parasites. They have to live inside a host cell. They're radically simplified. They lack mitochondria, peroxisomes, centrioles, many standard eukaryotic bits.
So why are they fungi then?
Molecular data seal the deal. Plus, crucially, their cell walls contain mitin and trehalose, key fungal signatures. And while they lack mitochondria, they have related structures called mitosomes. They represent extreme evolutionary streamlining.
If they're so simplified, how do they even get inside a host cell to begin with?
They have one absolutely incredible unique structure, the polar tube, or polar filament. It's coiled up tightly inside the spore under immense pressure. When the spore encounters a host cell, this tube ejects explosively.
Like a harpoon.
Exactly like a microscopic harpoon. It pierces the host cell membrane, and then the entire contents of the sporoplasm are injected through the tube directly into the host cytoplasm. It's an amazing invasion mechanism.
Unbelievable. And these are relevant to human health, too.
Yes, especially as opportunistic pathogens.
Yeah.
In individuals with weakened immune systems, species like Enterocytosome beanusi or Encephalidosome cuniculi can cause severe diarrhea, pneumonia, sometimes encephalitis, so clinically relevant despite their bizarre biology.
What a journey. We've gone from, well, caterpillar gold worth thousands to fungi manipulating amphibian electrolytes, fungi harboring bacterial assassins, essential plant partners, medieval poisons, deadly mushrooms, bat killers, and finally, these tiny intracellular harpoon artists. We've seen the yeast mold basics, the unique eukaryotic stage, decomposition, symbiosis. It's an incredibly diverse and impactful kingdom.
It truly is. And when you think about all these adaptations, the tough cell walls, those survival structures like sclerotia, the complex symbioses like with Burkholderia, the extreme specialization of pathogens like the WNS fungus, it really makes you wonder. So maybe something for you to think about is this. Given how good fungi are at surviving harsh conditions and forming these complex relationships, and given how tricky they are to treat as pathogens because they're eukaryotes like us, what selective pressures do you think push fungi towards becoming such specialized, almost stealthy pathogens compared to, say, bacteria? What makes them so uniquely suited for these roles?
Survival, specificity, and stealth. A lot to consider there. Thanks for joining us on this deep dive into the fungal world.